Good afternoon, students. So, today we'll continue with our chapter and we'll take up the next topic of this chapter, that is outbreeding devices. Now, as you are well aware, that most of the angiospermic plants, they possess the capability to inhibit germination of pollen grains and further growth of the pollen tube, so as to avoid inbreeding and to help the outbreeding. This chemical control of an individual plant is known as self-incompatibility also. This is achieved by interaction of chemical substances produced by the male gametophyte and the tissue of the style, like you can see in this diagram also, which I have displayed here on the screen. Yesterday what we talked about was landing of the pollen grain on the stigma, right? So now we are going to talk about what is going to happen after its landing. It has to germinate, or it will germinate, or it will not germinate. What will happen, right? That is what we are going to take up. So the stigma of a particular plant, when receives a number of pollen grains, they may be either from the same mating type or they may be from other plants. For example, if we take wind pollinated plants, so many different pollens of various plants will come and land on the stigma. Stigma is sticky, so it will stick there. So what has to happen after that? That is what we are going to discuss. So normally the functional pollens belonging to the right mating type will germinate on the stigma. They will develop a pollen tube and they will bring about fertilization. But if the pollen grains belonging to other mating types are falling on this stigma, what will happen? They will be discarded. They will not germinate. So if a pistil carrying functional female gametophyte gets pollinated with the right mating type of pollen grains but fails to set seeds due to inability of their gametes to fuse with each other, then this is known as sexual incompatibility. So this sexual incompatibility may be between the individuals belonging to different species, that is what is known as interspecific, or it can be between individuals of the same species, which is known as intraspecific, right? So do you get the difference between interspecific and intraspecific? The intraspecific incompatibility occurs between the members of the same species. Okay, do you get it? And this is the reason it is also known as self-sterility or self-incompatibility. The incompatibility involves many complex physiological, biological, morphological mechanisms which are associated with the interaction between pollen and tissue of stigma. It is controlled by genes. The interspecific incompatibility is controlled by more than one gene located on different loci of the chromosome. The intraspecific incompatibility is also controlled by genes with multiple alleles, which are known as opposition S alleles, like you see in the diagram. The incompatibility is achieved by genetically controlled physiological and morphological processes such as prevention of pollen germination, inability of pollen tube to penetrate into the stigma. Then it can be inhibition of pollen tube growth in the style, or failure of pollen tube to enter into the ovary or ovule, or it can be the failure of fusion of the male and the female gamete, or it can be last but not the least, abortion of embryo following syngamy or development of embryo endosperm incompatibility. So you get this thing that this incompatibility can be at any stage, from landing on the stigma up to the formation of the zygote and the endosperm. Now this self incompatibility, it can be divided into two morphological types, that is heteromorphic type and the homomorphic type. First we will take up the heteromorphic type. Heteromorphic type is the type in which two or more morphologically distinct mating types occur within a species, which can be recognized without a breeding test. You can see here, I'll just enlarge it, the pin type or the thorn type of this two different species. Here you can see the phenotypes of the plant, right? So it can be like this, that this is known as the heteromorphic type of self incompatibility. It can also be of the homomorphic type. In homomorphic type, the two or mating types within a species are morphologically indistinguishable. They require proper breeding tests. Based on the facts of pollen grain, the self incompatibility is divided into these two types. If the compatibility is controlled by the genotype of sporophytic tissue from which the pollen is derived, or the sporophytic tissue of stigma, then it is known as the sporophytic incompatibility. And if it is derived or it is due to the genotype of male gametophyte, that is pollen, then it is known as gametophytic incompatibility. Okay, so do you get it? The different types of incompatibilities we are talking about, that is the heteromorphic type and the homomorphic type. So in nature, this self incompatibility is to promote the outbreeding. It is to prevent the inbreeding and to promote the outbreeding. So they are also known as these devices which prevent the self fertilization or self compatibility. They are also known as the outbreeding devices. Now why this is necessary? This is also necessary in order to give more variation, so that if there is outbreeding, outbreeding is encouraged here, because you know that regular inbreeding will lead to inbreeding depression also. So all these processes or all these things which accompany or which promote self incompatibility are to benefit for the fertilization amongst different plants, that is they are the outbreeding devices. Understood? So now next part what we take is the pollen pistil interaction. Now what is this pollen pistil interaction? Pollen pistil interaction means that what will happen when the pollen will fall on the pistil or the stigma after pollination. The pollination does not guarantee the transfer of the right type of pollen. Do you get it? It guarantees the transfer of pollen, but whether it is of the wrong type or the right type, that it will not guarantee. Sometimes the pollen is of the wrong type. So it can be either from some other species or it can be from the same species, right? So the pistil, it has the ability to recognize the pollen, whether it is of the right type of the pollen or it is of the wrong type of the pollen, that is whether it is incompatible or it is compatible. So if it is of the right type, then the pistil accepts the pollen and promotes the post pollination events which will lead to fertilization. But if the pollen is of the wrong type, Then the pistil rejects the pollen by preventing pollen germination on the stigma or growth of the pollen tube in the style. So the ability of the pistil to recognize the pollen for its acceptance or rejection is the result of a continuous, you can say, a dialogue between the pollen grain and the pistil. This is a chemical dialogue. So this is mediated by chemical compounds or chemical components of the pollen which will interact with those of the pistil. So these interactions, they will lead either to the acceptance or rejection of the pollen grains. So this is a sort of chemical acceptance or chemical rejection. So this is for one thing. If it is rejected, it will not germinate. Even if it germinates, the pollen tube will not grow inside. You can see here these different types of the compatibility, incompatibility, whether it is going to grow. You can see in these three different cases like S1, S2, S3, these genes, and if they are not compatible, then it will not grow. Right here you see the different types of crosses showing S1, S2. This is fully incompatible. Then S1, S2 or S3, this is partially compatible, and if they are all different, that is S1, S2 is from one parent and S3, S4 are the alleles of the other, then it is fully compatible. So this is the way it will show whether it is compatible or not compatible. If it is not compatible, then it is finished there, but if it is compatible, then it will lead to the formation of a proper pollen tube, and this pollen tube will then grow inside. So after germination, if it is the right type of the pollen, then it will germinate on the stigma and will produce the pollen tube. You can see this in this diagram A. This is a photograph of the germination of the pollen grains. This is the in vivo germination, which shows the germinating pollen grains on the stigma. And this next diagram which you see, they are the pollen tubes growing through the style. These are elongated pollen tubes which are going through inside the style. And in the next diagram you can see that the pollen tube which grows fastest is entering here from the micropylar region for fertilization. Okay, is this clear? So once the contents of the pollen grain, they move towards the pollen tube grows through the tissues of the stigma and style, and it reaches the ovary. So when it reaches the ovary, here the generative cell may divide and then form two male gametes, and after that it will affect the fertilization. So the pollen tube, after reaching the ovary, it will enter the ovule through the micropyle and then enters one of the synergids to the filiform apparatus. Now this filiform apparatus is present at the micropylar part of the synergids. It guides the entry of the pollen tube. All these events from the pollen deposition on the stigma until the pollen tube enters the ovule are together referred to as the pollen pistil interaction. This pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. So here the knowledge gained in this area helps a plant breeder in manipulating pollen pistil interaction to get hybrids. So hybrids are formed naturally also, and they can be man-made also, what is known as the artificial hybridization, and we come to the next topic, that is artificial hybridization. So artificial hybridization, it helps in the crop improvement programs because we can select the parents, the male parent and the female parent artificially, then make a hybrid and get the desired characters. So in such crossing these experiments, it is important to make sure that only the desired pollen grains are used for pollination, and also the stigma has to be protected from contamination. Contamination here refers to landing of unwanted pollen. Do you get me? For artificial hybridization, two things are very important. One is that the pollen grains which we desire of the desired plant or of the desired flower are to land on the desired stigma, and secondly, the stigma or the female flower has to be protected from other pollen. That means unwanted pollen, no other pollen should land there. Now what are we to do for all these things? We are planning this pollination. So this pollination can be achieved by a technique which is known as the emasculation and bagging technique. Now here in this diagram you can see the process of emasculation, right? So what is emasculation? Now if there are this anthers, as well as, that means if a flower has both the male and the female part, then we remove the anthers from the flower bud before the anthers mature, right? This we do in a female flower. We remove the anthers and make it unisexual, make the flower unisexual. This is known as emasculation. You can see here that the anthers, they are removed by the process of emasculation from the female parent. Now in order to avoid the unwanted pollen to land on the stigma of this flower, these emasculated flowers, they have to be covered with a bag of suitable size, which is generally made up of butter paper, so that it prevents contamination of its stigma with the unwanted pollen. Now this process is known as bagging. So two terms now you are familiar with. One is emasculation, next is bagging. Now after bagging, here you can see this is the process of bagging. In bagging, this female flower is covered with a bag, usually which is made up of butter paper, and then when this female flower is mature, that means when the stigma becomes receptive, then mature pale pollen grains, they are collected from anthers of the desired male plant, and they are collected by means of a brush and they are dusted on the stigma. For this we remove the bag, we remove it temporarily, we dust the pollen grains on the stigma, and then we rebag the flowers till the fruit starts developing. And then what we do is we write different, we put a label or a tag on it where we write its information. This is what is known as tagging. So bagging and tagging, they are a simultaneous process. So this artificial hybridization basically involves the three processes, that is emasculation, bagging and tagging. Okay, this we'll also conduct in our practical class. We'll perform this in the practical class also. But if the female parent produces only this, that means it has only unisexual flowers, then one step can be skipped, that is emasculation, because there are no anthers present in the flower, so we don't need to emasculate it. They are to be bagged before the flowers they open. That means we bag them in the immature condition, in the bud condition only. We will put the bag on it, and when they become receptive, then we pollinate them and then we rebag them. So 3 steps are required for the bisexual flowers and 2 steps they are required for the unisexual flowers. Is it clear? So with this we complete the process of artificial hybridization, which involves the 3 different processes in a sequence, that is the emasculation, the bagging and the tagging techniques. Now we'll come to the last topic of today's lecture, that is the fertilization in these angiospermic flowers, what we call as double fertilization also. Here you can see the photograph of double fertilization on your screens. So now what do you understand by fertilization? Up till now what all we have studied the male flower, we have studied the female part of the flower, then how the pollen grains they get transferred to the stigma, that is pollination, and after pollination whether the female stigma accepts it or it rejects the pollen. Why and how it will accept or reject, that is dependent on a number of factors, and in case which is known as the pollen pistil interaction, and if it accepts it, then there will be growth of the pollen tube and then the fertilization. So this is what we are going to study now. Please be attentive. So fusion of the male gamete with the female gamete, that is the egg, to form a diploid zygote within the embryo sac, which is also called the female gametophyte, is what is known as fertilization. Now for this we will again start from the growth of the pollen tube. Right? We come down again to the growth of the pollen tube when it enters from the stigma into the style. So when it lands on when the pollen grains land on the stigma, they start germinating. Here they are sometimes 2-celled or sometimes they are at 3-celled stage. So here these pollen grains, which may be 2-celled or sometimes they may be 3-celled, they reach the receptive stigma of the carpel by the action when they land there, they germinate and then they start their growth. Now for germination, they will absorb the water which is present there and they will swell up, and then they release these they have some wall held proteins which will be released, and some other components also which are together known as the recognition factors. Now these recognition factors from the pollen and stigma, they will come close to each other. This is what we have discussed previously also for the acceptance and the rejection of the pollen by the stigma. So we are just sort of in a brief reviewing it again before we proceed further. So this mutual recognition or acceptance of the pollen grains determines its germination, and when it germinates it produces a pollen tube which grows into the stigma towards the ovarian cavity. Now the germination of this pollen tube occurs, or the pollen grain occurs, from the germ pore. Now do you remember where this germ pore is present? I told you before also. This germ pore is present where there is the missing of the exene, that is sporopollenin is missing there. They are the weak areas. They are 3 in number in the dicots and 1 in number in the monocot. So the initial growth of the pollen tube occurs at the expense of energy and food materials stored in the pollen tube. but subsequently derives nourishment from the cells of stigma and transmitting tissue of the style. The tube secretes exogenous pectinases, the enzymes and some other enzymes which are hydrolytic enzymes, which creates a passage for its entry. In most of the monocots and few dicots, the style is hollow. So the stylar canal is lined by glandular epidermis through which the pollen tubes grow on their way to the ovary. Okay. Now when it reaches the ovary, what happens here? When it reaches the ovary, now comes the next step, that is its entry inside. It can be through different places. Mostly which we see is through the micropylar end. This is known as the porogamy. Sometimes it can be through the chalazal end. Then it is known as chalazogamy. Example of this porogamy you can say like in lily flower, and in chalazogamy like in casuarina, or sometimes it can be between the two, that is the third type, which is known as mesogamy. This is present for example in cucurbita. So there are three different types, that is porogamy when it enters from the micropylar end, chalazogamy when it enters from the chalazal end, and mesogamy when it enters laterally. Of all these three, porogamy is most common, chalazogamy is less common, and mesogamy is very rare. So whenever we draw the diagram also, we draw of porogamy, which is most frequent. In porogamy, the tip of the pollen tube enters the micropyle, pushes through the nuclear tissue, and finally it pierces the egg apparatus. From where? From the embryo sac. Okay. In the embryo sac, it enters the egg apparatus. Now, when we talk about a typical ovule, we talk about this monosporic polygonum type of the embryo sac. Now it has three antipodal cells we discussed before also, then two synergids and one egg cell, and in the center is present the secondary nucleus. So the two synergids and the egg cell, they make the egg apparatus, and they are located at the micropylar end of the embryo sac. You can see here in the diagram also. If you cannot recall it just now, you can see in the diagram which is displayed on your screens. The synergids direct the growth of the pollen tube by secreting some chemical substances. The tip of the pollen tube enters into one synergid, and then it is penetrated. Here you can see in the diagram that it enters one of the synergids, and from here, when it enters here, you can see the synergid also starts disintegration. After penetration, the tip of the pollen tube enlarges and ruptures, releasing most of its contents, including the two male gametes and the vegetative nucleus, into the synergid. So one male gamete fuses with the egg to form the zygote. The process is called syngamy or generative fertilization, and the diploid zygote finally develops into the embryo. The other male gamete fuses with the two polar nuclei, which are also known as the secondary nucleus, to form the triploid primary endosperm nucleus. This process is known as triple fusion or vegetative fertilization. Now these two acts of fertilization constitute the process of double fertilization. That is why we call the fertilization in angiosperms as double fertilization. Do you get it now? Why we say the word double fertilization in angiosperms? Because the two male gametes which are present in the pollen grain, one of them fuses with the egg cell to form the zygote, and the second one forms the primary endosperm nucleus. It fuses with the two polar nuclei. So the ploidy here is also different. In the zygote, the ploidy is 2n, and of the endosperm, the ploidy is 3n. So this is where we stop today. I hope you understood, and whatever difficulties you have, you can text me, and I'll try to make those difficulties, remove those difficulties, and try to make them clear to you. So we'll take up the post fertilization events tomorrow, and we'll meet again tomorrow at the same time, that is 2 o'clock. Okay childrens, then now you please go through this lesson and do your assignment which is displayed in your class, and turn in your assignment at the right time. Please don't forget to mark your attendance. Thank you.